our next speaker in the session, Phuong Tong, who is a medical student at the University of Washington. She also participated in the 2019 cohort of the NIH Medical Research Program, and she will tell us about cortical ripples in humans. Please take it away.
Thank you for that introduction, and thank you everyone for listening in. So I will talk about our study on ripples in human cortex and how it reflects the spectrum of synchronous spiking activity across spatial scale. So first, I want to start off with a framework for understanding the mechanism for cognition, which we can think of as these transitions between asynchronous and synchronous activity, whereby at one end, you have neurons that represent different information and have distinct spiking. And then there are periods in which neurons that have shared representations communicate by spike synchronization. And these perform computations that are necessary for cognition. So we study oscillations in the brain from local field potential recordings and from intracranial EEG recordings in order to assess the level of synchronous activity in the brain that is important for cognitive processes. So in particular, we've looked at 70 to 200 hertz frequency band, which I will refer to as the high frequency activity. And we've seen that we can detect synchronous activity that it underlies attention and successful memory retrieval and encoding. And then more recently, we've also found that the 80 to 120 hertz band, in particular in humans, which I will refer to as the ripple band, is also important in successful memory retrieval. And we know from vast rodent literature that these ripples can be detected as events of synchronous activity. And similarly, we've done this in humans, but we use these arbitrary detection thresholds, and it's still unclear whether these ripple events can represent the synchronous activity in this spectrum of synchronous activity. And also we measure this ripple signal in macroscale recordings, and it's unclear if these macroscale recordings at the ripple band actually represent this spectrum of synchronous activity. So we explore the ripple signals in human cortex further. In 21 patients with intracranial EEG recordings, we asked to what extent do ripples resemble high frequency activity at the macroscale? And in 6 participants with simultaneous intracranial EEG and local field potential recordings, we asked how are ripples and spiking activity related across spatial scale? And we asked to what extent are synchronous spiking activity and ripples rhythmically modulated across spatial scale. So we measure local field potentials from microelectrode arrays, and we can compare these microscale signals to macroscale signals from intracranial EEG context nearby. And so during these recordings, we asked the patients to participate in a paired associates verbal memory task in which there are 2 periods. The first encoding period, we cue the patient to form a memory when they're presented with a word pair from a list of word pairs. And then we have an interval period in which we give math distractors, and we follow that with a retrieval period in which the patients are cued to recall a memory when they are presented randomly with 1 word from the previously studied word pairs. And then we take the recordings offline and we can assess the ripples and the high frequency activity at the macro scale and at the microscale and compare it to underlying spiking activity. And we can assess how these signals are related during memory retrieval. And so first, in order to compare ripples and high frequency activity, we decompose the intracranial EEG signal using a wavelet transform. And from the time frequency plots as shown on the bottom, we observe these discrete periods in which there's high activity at the high frequency range. And specifically, there seems to be high activity in the 80 to 120 hertz range, or the ripple band. And we can use the ripple detection thresholds that we previously published on to identify these discrete narrowband events. And so we can compare the high frequency activity and the rate of ripples prior to vocalization during retrieval, as indicated by the time 0. And from the time frequency plot on the left, we can see that there's an increase in high frequency activity just before vocalization across all trials. And we can similarly see an increase in the rate of ripples just before vocalization, as seen in the raster plot underneath. And when we separate out correct trials and incorrect trials, we see that specifically successful memory retrieval exhibits increased high frequency activity and increased rate of ripples. And further, when we compare the change in high frequency activity and relate it to the change in ripple activity, we find that these measures are correlated in memory relevant brain regions. So specifically in the medial temporal lobe and in the temporal association cortex, or the anterior temporal lobe. And so these findings suggest that ripples are a measure of synchronous activity that's represented by high frequency activity changes resolved in time. And then next, we wanted to know within temporal association cortex, how are signals across spatial scale related? And so we make pairwise comparisons across spatial scale, and we modified the ripple detection threshold in order to assess smaller events. And so first, we compared spiking activity and microscale ripples, and then spiking activity and macroscale ripples. And just from looking at ripples across spatial scale, we see that when these ripples occur, we also have more spiking events occur. And so when we computed the correlation between ripple amplitude and spike rate, we found a significant correlation for both the microscale and macroscale across patients. And this is demonstrated in the middle plot, where each data point represents the correlation for one patient. We similarly looked at individual macroscale ripple events and characterized the spike content for each ripple event by looking at the proportion of spiking units. And we found that macroscale ripples reflected the number of spiking units. We further compared macroscale ripples and microscale ripples. So this time we just focused in on the ripples. And so from looking at the signals simultaneously, we found that when there were more microscale ripples, the macroscale ripple tended to be larger. And so when we computed the correlation between the macroscale ripple amplitude and the proportion of microelectrodes with ripples, we found a significant correlation, suggesting that macroscale ripples reflect the sum of microscale ripples. And then we further observed that when the microscale ripples were more aligned, these were associated with larger macroscale ripples. So we wanted to measure the synchrony of microscale ripples using pairwise phase consistency. And so to the right is a diagram showing how we assess synchrony of microscale ripples using these pairwise phase differences across the ripple signals in the microelectrodes. So from that analysis, we found that macroscale ripples not only reflect the sum of microscale ripples, but also their synchrony or alignment. And so further, we looked at the relation between spiking activity and microscale ripples. So this time we assessed the LFP or microscale ripple amplitude correlation with the spike synchrony as assessed by, again, the pairwise phase consistency, and found a significant correlation between these measures, suggesting that microscale ripples reflect the synchrony of spiking activity. So altogether, this suggested that macroscale ripples reflect synchronous microscale ripples, and microscale ripples reflect synchronous spiking activity. And then next, we wanted to know to what extent are synchronous ripple and spiking activity rhythmically modulated. And so we computed the spike triggered average as shown on the left top and the ripple triggered average as shown on the left bottom. And we found that these events appear to be coherent with low frequency oscillations. When we computed the pairwise phase consistency of these events across frequencies from 2 Hertz to 200 Hertz, we found that the spiking activity and ripples are modulated by low frequencies around 3 Hertz. And so further, we wanted to know now with synchronous spiking activity and ripples, so events that occurred coincident with ripples, how were these activity modulated? And so again, we computed the pairwise phase consistency for these events during ripples, and we found that they were modulated by low frequencies at 6 and 11 Hertz. And so altogether, this suggests that both ripples and spiking activity across spatial scale are modulated by low frequency oscillations. And so again, to come back to our framework for understanding the mechanism for cognition, we can think about ripples as these periods of rhythmically coordinated communication that occur during these continuous periods of distributed computations. And so when we detect ripples, we indeed can detect these periods of synchronous activity along the spectrum of activity. And specifically, when we detect larger ripples, that may indicate more shared representations across neurons. And so overall, our findings would suggest that these ripples measured at both the macroscale in the intracranial EEG recordings and the microscale in the local field potential recordings reflect transient engagement of synchronized spiking activity during successful memory retrieval. With that, I'd like to thank my lab, Dr. Zaghloul's lab, and I've left my contact information if anyone has questions offline. Thank you.
Awesome, thank you. So I'm by no means an expert in this field, but my understanding from the rodent literature is that during an event, there's some spiking activity that happens in a neuronal ensemble, and then during a ripple, this same spiking activity is often sort of replayed, you know, to enforce these memory events. Since you guys are actually recording spiking activity in humans, which I have to say is absolutely phenomenal, do you guys see a pattern like this? Can you tease something like this out in the human data?
Yes, so there has been a paper published by another member of the lab that has found that there is a reinstatement of these spiking events or the sequence of spikes, and that they were correlated with these repeats in the ripples that occurred during encoding and also similarly occurred during retrieval.
That's awesome. That's really cool work.
Thank you for the question.
All right. Thank you very much. I think in the interest of time, we will